Degenerate primers. It is another type of primer that can also be used for in vitro amplification of the DNA. It depends upon our requirement that what DNA sequence we have focused and from where we have deduced the sequence of the nucleotides to synthesize the primer. So as far as its definition is concerned, degenerate primers, basically it is a mixture of primers where all the fragments in the primer they have more or less similar sequence but varied at one or a few position. So such a mixture of primer it is called as the degenerate primer. क्योंकि बाज़ दफ़ा होता है कि इसका सीक्वेंस तो एक ही होता है लेकिन बाज़ दफ़ा हमें ज़रूरत होती है कि कुछ बेसिस जो हैं वो रिप्लेस हो जाएं. लाइक सपोज करें कि अगर पोजीशन 10 पे एडानिन है तो दूसरे फ्रेगमेंट में एडानिन की जगह जो है वो गुआनिन हो सकती है. उससे नेक्स्ट फ्रेगमेंट में जो है वो पोजीशन 12 में अगर गुआनिन है तो पोजीशन 12 पे थाइमिन हो सकती है. तो इस तरह से ये जो डिजेनरेट प्राइमर्स हैं ये ऑल दो मोर और लेस इनका जो सीक्वेंस सिमिलैरिटी जो है वो सेम होती है एक्सेप्ट एक दो पोजीशन पे वो वेरी कर रहे होते हैं. नो व्हाट इज द रीजन दैट व्हाई वी नीड टू सिंथेसाइज primers in the form of mixture and why we call it them as a degenerate primer. बात सिचुएशन में होता है कि हमारे पास डायरेक्ट न्यूक्लियोटाइड का सीक्वेंस नहीं होता और हमने इंफॉर्मेशन जो है वो किसी और मॉलिक्यूल से ली होती है लाइक फ्रॉम द सीक्वेंस ऑफ द अमीनो एसिड्स कि हमारे पास एक अमीनो एसिड का सीक्वेंस एक प्रोटीन है और उस प्रोटीन में अमीनो एसिड का सीक्वेंस देखते हुए हम ये प्लान करते हैं कि इससे हम न्यूक्लियोटाइड का सीक्वेंस डिड्यूस करें या डिकोड करें. तो after decoding we may have the knowledge that this particular codon it may be involved in the incorporation of a particular amino acid. So in this way we can deduce the sequence of the nucleotide from the information that is available from the sequence of the amino acid. But during the synthesis of such degenerate primers we should account the degeneracy of the codon. Degeneracy, you have the idea that for many amino acids there may be more than one codon that are there for the incorporation of a particular amino acid. And in order to synthesize the primer, we should also take into account this process of degeneracy कि एक पर्टिकुलर जो अमीनो एसिड है इसके लिए एक से ज़्यादा कोडॉन हो सकते हैं. तो इसको अकोमोडेट करने के लिए हमें प्राइमर का मिक्सचर जो है वो तैयार करना पड़ता है. Now degenerate primers they have different advantages or application because we can search gene families or related gene families, or if we want to find homologous genes in different species, even then we can use the sets of degenerate primers. This table showed the degeneracy of different codons and how many codons they are required for different amino acids. And on the basis of number of codon per amino acid, we can categorize them into 6 major classes. Like in case of first column, the one codon it is required for methionine or tryptophan, and there are number of amino acids that have 2 codons. In case we can quote the example of cysteine, asparagine, glutamic acid, and so on, like the tyrosine. And isoleucine, it is encoded by 3 codons. And similarly, 4 codons are required for alanine, glycine, proline, threonine, valine. And even there are some amino acids like leucine, arginine or serine that have 6 codons. So we can say that for the sixth column, where 3 amino acids are there, the degeneracy, it is relatively very high. And from this information, we can decode the sequence so that we have the idea that this amino acid sequence, it is required for the synthesis of degenerate primers. And we should keep the degeneracy issue in our mind while attempting the synthesis of degenerate primers. And this table, it indicates that how we can synthesize, or how the degenerate primers, they can be represented. So this is the standard nucleotide coding system during the synthesis of degenerate primers. मिसाल के तौर पर अगर हम एडानिन इनकॉर्पोरेट करना चाह रहे हैं तो उसका जो बेस की जो रिप्रेजेंटेशन है वो एडानिन ही होगी. In case of cytosine, it is cytosine in the strand, G for guanine and T for thymine. And then if we want to synthesize the mixture of primers, no different symbols according to the coding system, they can be given. Like if in the sequence R is given, it means that it contains a mixture of two fragments. Like A may be there, and in the second fragment it may be represented by the guanine. And similarly, Y, it is represented by C and T bases. In addition to that, the same position, it may be represented by 3 nucleotide bases. Like in case of, if the letter H is there in the primer sequence, it will indicate the presence of either adenine, cytosine, or even thymine at the single position. So the 3 fragments we have to synthesize, and sometime if it is given the letter N, so it may be represented by 4 different nucleotide bases, like either at this particular position adenine is there, cytosine, guanine, or thymine. So we have to synthesize 4 different fragments in order to cover the degeneracy of the genetic code. So in this way we can say that it is a mixture of the primer that is used for the in vitro synthesis of the DNA. Now, with reference to primers or degenerate primers, now we have the idea that primers, they are mainly used as a starting point for the DNA synthesis. They provide free 3' hydroxyl group for in vivo or in vitro DNA biosynthesis.